cloud. And unless otherwise requested by the speaker, there'll be this, this talk, like the others, are being made available on the YouTube channel for public viewing. And so I'll go ahead and paste some of this information into the chat box. And if you just have any questions along the way, technical or otherwise, you can message me via the chat box and I'll try to help you out. So today I get to introduce our speaker who is going to round out the series for us, who's my friend and colleague, Dr. Nicholas Dove. Dr. Dove is a postdoctoral associate in the Biosciences Division at Oak Ridge National Laboratory, which is in Tennessee. And his research focuses on generally better understanding mediators of microbial community assembly and how this influences plant and ecosystem health. Dr. Dove earned his doctorate from the University of California, Merced's Environmental Systems program, working with Dr. Steve Hart, who is the same major professor that I worked with for my PhD. And today he's going to be talking with us about the effects of fire on the soil and plant microbiome. So Dr. Dove, I know it's getting late over there where you are on the other side of the country, and I just want to say thanks so much for taking the time to join us today. Super glad for the opportunity to hear about your work, and why don't you go ahead and take it away?
Thanks, Chelsea, and thanks everybody else for joining today and the opportunity to share some of my science. But before I do, I'd like to tell a short story. Now, I've been interested in fire for almost a decade now, and interest goes back to my time working for the Bureau of Land Management in California and Wyoming. And it was there I was first introduced to wildfire. And something I found interesting is that certain areas recovered successfully and quite rapidly after fire, while others had a more stunted regeneration and were invaded by non-native invasive plants. And when I asked my supervisors why this was, I wasn't totally satisfied with answers. I was told it had something to do with the soils, but it was generally unclear. And like I said, this vague answer wasn't necessarily satisfying and really motivated me to go to grad school and pursue a career in research. And since then, my interest in wildfire and how it shapes ecosystems has only grown. But on the news, fire is generally portrayed as a devastating catastrophe. And in many ways it is in terms of the impacts on human life and property. So I don't want to minimize this, but fire is also a fundamental ecological process that has influenced Earth's ecosystems for millennia. As you can see, at any given time, millions of hectares on Earth's surface are burning, and this results in distinct fire-adapted ecosystems, such that a world without fire would look drastically different. However, even though fire is essential in structuring many of Earth's ecosystems, it is important to recognize that fire regimes, or the way in which fires burn, are changing. So for a variety of reasons, most of which are human-linked, fires around the world are increasing in size, severity, and frequency, which is significant because ecosystems that have been adapted to certain types of fire are becoming less and less adapted. And unfortunately, this threatens the sustainability of many terrestrial ecosystems, prompting even the UN's environmental program to ask the question, are these megafires the new normal? As I alluded to before, these are real-life examples of what ecologists call state changes, where a disturbance or multiple disturbances are so severe that the ecosystem, instead of recovering to its previous state, becomes an entirely new ecosystem. And this is generally considered to be a negative process, which has negative consequences for numerous ecosystem services. And as a microbial ecologist, I'm interested in the way that microorganisms impact plant health and soil quality. The science behind the importance of microbes to plants and ecosystems is well established. So my question is, can microorganisms therefore mediate ecosystem recovery and preserve some of these services? For the past few years, I've been trying to understand how microorganisms respond to and mediate the effect of fire on Earth's ecosystems. And I, among others, have begun to answer questions ranging from basic to applied. We want to know, what is the impact of fire on these microbiomes? How long does the impact last? How then do these ecosystems mediate recovery? And finally, can we use microorganisms in such a way that leads to desirable outcomes for humanity? Now, to answer this first question, what is the impact of fire to the plant and soil microbiomes? we sampled across a burn severity gradient in a prescribed fire or an intentionally set fire in the mountains of central Utah. And there we sampled 9 plots of each treatment where we destructively collected 3 less than 1-year-old aspen saplings and the associated soil. And as you can see, the aspen recovery was quite rapid, as this was only 3 months after the fire. And I'll also mention that this work was recently accepted in New Phytologist, and this picture that I took was also selected for the cover of that issue. And from each sapling, we sampled the microbes living in or on the surface of the leaves, stem, the root, and the rhizome. And the rhizome is the coarse root that connects these clonal aspen plants, as well as the rhizosphere, the soil that's directly associated with the root and bulk soil. From these samples, we sequenced 16S and ITS amplicons, or certain barcodes for that describe the bacteria and fungal communities. And we set out to answer the following questions. How do the soil and plant microbiomes respond to fire? We hypothesize that the bulk soil will have greater differences in microbial community composition than the plant-associated communities, highlighting the strong control of plants over their microbiome. Then going a step further, what drives the microbiome response to fire? We hypothesize that fire-induced changes to soil chemistry and the associated changes in plant tissue chemistry will explain a significant proportion of the microbiome composition. And finally, how does fire disrupt microbiome recruitment? We know from previous work that fire can sterilize or at least decrease the abundance of microbial biomass in the upper soil layers. Assuming this is true in our case, we hypothesize that soil will decrease in its role as a source of microbial taxa for the aspen microbiome. First, looking at community composition, consistent differences began to emerge. And here I am showing multidimensional representations of the archaeal and bacterial and fungal communities among different sample types, where each point represents a sample. In these figures, closer points represent more closely related samples. And the variation explained by each axis is given in parentheses. Finally, the asterisk next to each sample type indicates if indicates a significant fire response. Now, as you can see, the impact of fire microbiome composition depends on sample type, but that was pretty consistent between prokaryotic and fungal communities. For the most part, differences were leaf and soil samples, and there was a weak but significant impact of fire on the prokaryotic rhizome microbiome. Taxonomically, these differences emerged at the phylum level, and here I am showing the relative abundance of important prokaryotic taxa. In the leaf, you can see that we have increasing firmicutes and decreasing actinobacteria. And while less apparent in the soil microbiomes, there's a shift from chloroflexy towards firmicutes as well. Now, changes in the fungal community composition with fire also emerged at the phylum level, particularly in the leaf and bulk soil. There was also an increase in Basidiomycota relative to Ascomycota, and in the rhizosphere, fire was associated with an increase in fungal phyla, such as Mycorrhizal and Mycorrhizal. We're also interested in the functional significance of these changes in the microbiome. And for this, we focus our analysis on pathogens, which negatively affect plant health, and mycorrhizal fungi, which are mutualistic symbiomes that can positively impact plant health. And for those of you unfamiliar with mycorrhizae, these are fungi that expand the plant root networks and increase nutrient uptake for the plant in exchange for plant photosynthase. In the leaf, we saw an increase in the relative abundance of potential fungal pathogens with increasing burn severity, particularly the genera Claudosporum, Alternaria, and Arocyphe. And below ground, while we did not see a shift in the relative abundance or richness of mycorrhizal sequences, we did see a shift in the mycorrhizal community. Interestingly, fine root patterns differ from the soil microbiomes. In the fine root, fire results in increased Wilcoxina and decreased Russula, while fire increased the relative abundance of Russula in the soil microbiomes, and at least in the bulk soil, decreased the amount of Wilcoxina. And this highlights that mycorrhizal associations here are more specific than who's directly near the plant in high abundances. So coming back to our original hypothesis that greater differences would occur in the soil microbiome compared to the plant. This hypothesis was generally supported. However, we did see large changes in the leaf microbiome with fire. And these changes in the leaf for both prokaryotes and fungi were stronger than the soil response, which I thought was pretty surprising considering that these leaves emerged after the fire. So these leaves, unlike the soil microbiome, were never directly exposed to fire. And so this brings us to our next question. What is driving the response in these different sample types? Consistent with our hypothesis, both soil and leaf chemistry change in response to fire. For instance, soil pH, consistent with many previous studies, increase with burn severity. And what happens is the combustion will release a lot of base cations from organic matter, resulting in these high pHs. Soil carbon and nitrogen, while qualitatively lower with fire, were not significantly different than the unburned site. And this wasn't too surprising. While fire likely combusted a lot of soil carbon and nitrogen, charred remains from above ground carbon and nitrogen sources likely entered the soil solution. And nevertheless, while I can't definitively show this, I can imagine that the chemical composition of these carbon compounds changed with fire, where you have an enrichment of polyaromatic structures. And while I am not showing these data here, I also know that the inorganic nitrogen in our sites actually increased with fire, which is also well documented in other studies. In plant tissues, the largest differences in elemental composition were in the leaf, which sort of corroborates what we found in their microbiome. The leaf was significantly enriched in nitrogen and had less potassium. And then now, to look for correlations between the soil and leaf chemistry on the microbiomes, distance-based redundancy analysis. Similar to PCOAs, which was the previous plots that look like this that I showed, I'm showing another multidimensional representation of the microbial community. But instead of maximizing variance explained on two axes, I'm maximizing the variance loaded onto the plant and soil chemistry data sets. And here I'm only showing the leaf and soil microbiomes because these were the ones that differed the most by fire. And on the soil side, I lumped together the rhizosphere and bulk soil. And I should also mention that the direction of each arrow shows how the samples relate to that variable increasing. And the length of each arrow shows the relative predictive strength of that variable. Consistent with numerous studies from soil microbial ecology, we find that pH plays a strong role in structuring these microbiomes. And at least for prokaryotes, we see that carbon and nitrogen are associated with the differences between the rhizosphere and bulk soil communities. In the leaf, at least for prokaryotes, nitrogen is one of the strongest correlates of the microbiome composition. However, looking at all these ordinations, you can see that we are explaining at most 15% of the variation in microbiome composition. And there are a few explanations for this that I will address shortly. But first, coming back to our hypothesis that fire-induced changes in soil and plant chemistry would impact the microbiome, this hypothesis was somewhat supported. Yes, we did see correlations between pH in the soil microbial community and leaf nitrogen in the leaf microbiome, but there was still a lot of unexplained variance. And I think this could be due to a couple of different things. One, our characterization of soil and plant chemistry were far from comprehensive. We only looked at elemental concentrations, which are useful, but don't necessarily explain the molecules a microbe would sense. We know from other studies that aspen leaves post-fire are enriched in phenylglycosides to minimize insect debris. And these molecules have also been shown to influence the microbial community. So I think this really underscores the fact that we need to complement our microbiome work with metabolomics if we're really going to understand what structures the plant microbiome. Secondly, there are likely other biological and physical processes going on that would explain these differences. For example, after fire, the forest canopy is burned, and the amount of solar radiation reaching aspen saplings increases drastically. While we do not have these data, I imagine this probably plays a role of who can survive in a leaf. And finally, this is only 3 months after fire. Who survives in the soil is partially random and partially biological. I think much of what we are seeing is a legacy from the direct effects of fire. Which brings me to my third hypothesis, that this selective soil sterilization legacy And soil sterilization, I'm saying with air quotes here, because we know that fire doesn't sterilize the soil, but it does reduce microbial biomass. But anyway, that this soil sterilization legacy could influence the relative dominance of vertical versus horizontal microbiome transmission. So just like the human gut microbiome can be colonized horizontally through what we ingest and who we interact with, or vertically through the mother, plants that propagate clonally, like aspen, also have both horizontal and vertical modes of microbial transmission. So the question is, does fire impact the dominance of microbiome transmission? And we hypothesize that with decreased microbial abundance in the soil directly after fire, that vertical transmission would increase in dominance. And to do this, we use FEAST. And this is a microbial source tracking algorithm, similar but more accurate and faster than the widely used source tracker algorithm. And by using the relative proportion of different sequence variants, it develops Bayesian mixing models to determine the relative contribution of the microbiome at from different sources. And before I show you the results, let me mention that these are models, which need to be empirically tested. However, I think they're useful then in determining if we're thinking about microbial transmission correctly. And our hypothesis that fire-induced soil sterilization would increase vertical transmission was somewhat supported by the data. So here I am plotting the percentage of each source, which are the rhizosphere, bulk soil, and rhizome, in each sink microbiome, which are the stem, leaf, and fine root across both amplicons and burn severities. And first thing I'll bring your attention to is that these percentages for each sample, these 3 bars clustered together, never equal 100. And that's because the model was unable to classify much of the source taxa. But that being said, of the taxa it was able to classify, our hypothesis was somewhat supported. For prokaryotes, at least in the stem, vertical transmission increased with burn severity. And for fungi, in the above ground microbiomes, horizontal transmission decreased with burn severity. Nevertheless, the lack of source attribution minimizes what we can really say about this. But I think this shows that disturbance can affect patterns of transmission, and that these patterns may be important in plants like they are important in humans and other animals. So how does this research fit in with our previous questions? Well, we found that the plant microbiome was for the most part less sensitive to fire, with the exception being the leaf, and that these changes are partially due to differences in soil and plant chemistry and microbial transmission with fire. But these next 2 questions we were not able to answer with the current study, but I will now present some of my PhD research on the soil microbiome to show that these results can be long-lasting and may impact ecosystem recovery. To determine how long the impact of fire lasts, we use the Sierra Nevada fire chrono sequence. And the Sierra Nevada fire chrono sequence is a time for space substitution technique where we use fires of different ages to track the recovery of a stand after one fire. And we have fires ranging here from 1973 to 2013. And using our selection criteria, we made sure to minimize the effect of location. Interestingly, we found that the impact of fire is long-lasting. So even though the disturbance itself can be over in a matter of days, its legacy can last for decades. Even 25 years after disturbance, the soil microbial community was still significantly different from the late successional site, which hadn't seen fire in over 115 years. And taxonomically, here I'm showing the relative abundance of different phyla. And there was this monotonic pattern with time since fire, such that the proportion of actinobacteria decreased and proteobacteria increased. And this, I think, gives us some confidence in our study design. A common concern with space for time substitution is there might be some noise other than the environmental gradient in question. So because we see gradual changes in taxonomy, makes me think that we are seeing the effect of time since fire. Similar to the plant study, we were interested in what specifically is causing these
I think we might have lost Nicholas here. Let's see if we can get him back. Hey.
I'm back. I don't know.
That's all right. It made sure we're all awake. So yeah, do you want to go ahead and screen share again and keep going? Awesome.
Let's do it. Yeah. That was weird.
What is a virtual presentation without a?
I know,
like
exactly.
Let's
see.
Okay, and I'm gonna let's see. I'm gonna go ahead and spotlight you again.
Okay.
There we go. So yeah, I lost you sort of when you started this slide. So if you want to,
yeah, I'll just start this slide from the top.
Awesome.
So similarly to the plant study, we were interested in what specifically is causing these changes. And to do this, we investigated the different types of fundamental ecological assembly processes. Now, fundamental ecological assembly can be characterized as either stochastic, meaning who colonizes and grows is primarily a product of chance and priority effects that favor early colonizers, or deterministic, meaning who colonizes and who grows is primarily a product of selection. And furthermore, selection can be broadly broken into either homogeneous selection or variable selection. And homogeneous selection creates homogeneous communities based on the environmental gradient, where communities are more similar, and heterogeneous communities, depending on the microbes and environmental gradients, can be more dissimilar due to this selective factor. And to characterize these assembly processes, we used a null modeling approach that measures the deviance in taxonomic and phylogenetic community structure from a null community model. And we found that, in part, time since fire resulted in greater homogeneous selection. So early microbial communities are relatively more stochastic, but become more selective with time. And selection is caused partially by changes in above ground vegetation and soil chemistry. While fire was the dominant factor in explaining microbial community composition, there was still a smaller but significant main effect of vegetation. We also saw large changes in soil chemistry among the sites, which also explained a large proportion of the variance. For instance, soil carbon in the greater than 115-year site was almost double all other sites, which underscores the majority of these fires and the lack of resilience in this pool. And this is a topic for another conversation, but I thought this was a really significant finding, considering a global change context where we want to store soil carbon. We also binned shotgun metagenome sequences into metagenome assembled genomes, or MAGs. And these represent discrete genomically resolved biological units, similar to genomes sequenced from microbial isolates, to investigate which microbes thrive post-fire. We found that the classification of early and late successional MAGs, based on their normalized abundance with time since fire, was taxonomically resolved, similar to our amplicon results, which showed actinobacteria responding positively directly after fire, and proteobacteria becoming more abundant with time since fire, with a few exceptions at the order level. But the question is, why are these genomes succeeding directly after fire, and what genes allow them to succeed? But first, let's begin to understand what the post-fire soil condition is. First of all, post-fire soils are enriched in ash, which is similar to biochar. And from here on out, I'll call this pyrogenic carbon, or Pi-C. And what happens is that incomplete combustion of organic matter causes this pyrolized, the incomplete combustion pyrolyzes organic matter, such that these condensed polyaromatic structures begin to form. And this structure is not easy for a microbe to degrade. And in fact, many do not possess the genes to produce enzymes that can degrade this. Thus, if you're a post-fire microbe that can degrade pyrogenic carbon, you are likely apt to succeed. Second of all, post-fire conditions are quite stressful in terms of solar radiation and drying out, since there is no longer an organic layer protecting the soil surface. So again, if you can tolerate moisture and solar stress, who are likely apt to succeed. And finally, directly after fire, microbial abundances are low. So if you are a fast grower, you can likely get a foot in the door in these ecosystems. Therefore, we expect that these early successional microbes can degrade pyrogenic carbon, are stress tolerant, and are fast growing. Now, starting with the ability to degrade pyrogenic carbon, early successional microbes were far more likely to possess genes encoding for oxidative enzymes necessary for aromatic degradation. And here I'm showing the normalized enrichment of different aromatic pathways, with red indicating greater likelihood of presence. Therefore, you can see in many of these that the early successional microbes possess a lot more of these aromatic degradation pathways. And I think this has some ecosystem level implications. As I said before, these compounds are difficult to degrade and are generally thought of as being useful for carbon sequestration. But here, I think that we see that, in fact, this carbon pool may be quite susceptible to decomposition if some of these early successional microbes are present. We also found that early successional microbes were far more likely to have genes encoding for stress tolerance. Ectoin and mycorrhizal are produced to alleviate osmotic stress within the cell, and the sigma B stress response is a pretty cosmopolitan stress response where it responds to heat, water, and radiative stress. So a lot of these early successional microbes are also very stress tolerant. Finally, we used GRID to measure in situ microbial growth rate. And GRID is an algorithm which is published in 2018, is similar to the widely used IREP algorithm to measure microbial growth rates bioinformatically, but it can be used at much lower coverage levels. And the basic idea is that growing bacteria have higher amounts of DNA at the origin ends of their genome, where they're replicating compared to the terminus ends. And so it uses this coverage-based analysis to determine growth rates. And so we found that on average, there were no differences between the average growth rate of early and late successional mags, but we found that the greatest growth rates occurred during mid-successional time points. And this is a little different than what we expected, being that early successional genomes would have higher growth rates. But I think this makes sense considering the greater carbon and nitrogen availability in these mid-successional sites that likely spurred microbial growth. Therefore, our hypothesis that early successional microbes degrade carbon, pyrogenic carbon, are stress tolerant, and are fast growers was only partially supported. We now see that they can degrade pyrogenic carbon and are stress, but are not fast growers, which contradicts earlier predictions that early successional microbes grow quickly. Now, on kind of a larger scale, to look at how the microbial community as a whole influences ecosystem recovery, we looked at functional genes in our metagenomes. And here I'm presenting differences in carbon degradation potential using the KZ gene database. And directly after fire, we see an increase in KZ abundance. These are these carbon degrading genes, especially genes associated with cellulose degradation, and that this high abundance declines with time since fire. Alternatively, potential for lignin degradation increases with time since fire, suggesting a changing microbial metabolism, which may influence carbon storage on these landscapes. We also saw an enrichment of nitrogen cycling genes directly after fire, particularly that of nitrogen fixation and ammonia oxidation, which is the rate-limiting step of nitrification, allowing for nitrogen uptake by the plant. And combined, these gene abundances correlated with overall inorganic nitrogen availability.
Uh-oh, I wonder if we lost Nicholas again. Let's see. See if he can hop back on here. Let's give him a second to figure it out on his end. Sit tight, everyone. Hopefully he'll hop back on in just a second. Seems like he has some connection issues. I hope everyone is having a great Earth Day. In the meantime, happy Earth Day. Let's see. I'm going to go ahead and send him a text message and see. Oh, here he is. Let's see. Hey.
Sorry about that. I don't know what's going on.
Okay, they have pretty spotty service out there in at Oak Ridge, huh?
I guess so. I don't know. This doesn't normally happen, but I just turned my internet off and turned it back on. And so hopefully this won't happen again in the final stretch of the presentation.
It's all good.
But yeah.
No worries. No worries at all.
Yeah. How embarrassing.
It's okay. I think we can all, you know, I think we've all sort of probably had similar experiences in the past. Yeah. So I'm sure there's a lot of empathy coming your way right now.
Yeah, fair enough.
Yeah.
So I don't know how much folks heard about the
I'd start from the beginning with this slide.
Yeah. So we also found that fire negatively impacts both the diversity and colonization potential of mycorrhizal fungi, which are critical plant symbionts, and many plant species, including pines, will simply not grow to maturity without their presence. And from the Sierra Nevada fire chrono sequence, the richness or number of OTUs, and you can think of this as species, increased with time since fire. And I'm also presenting data from a somewhat recent meta-analysis that we conducted, which showed that potential mycorrhizal colonization patterns, on average, don't recover until 12 years after fire. And together, these findings suggest that lack of mycorrhizal inoculum may stunt regeneration of native vegetation, allowing for the dominance of rhodoral non-mycorrhizal plants. Now, coming back to our spectrum of questions, we know that at least in the soil, recovery of microbial communities can take decades. And this can result in long-term changes in microbial functionality and how microbes interact with plants. So that if post-fire we have negative effects on the mycorrhizal community, which we see across 3 of these studies, and if fire increases fire leaf pathogens, plant survivability in post-fire environments may be jeopardized. Furthermore, alterations in the carbon and nitrogen cycle will likely affect the ability for ecosystems to sequester carbon and store nutrients for plant growth. So as I mentioned before, this research is ongoing. So we haven't yet begun on the more applied science, but with my remaining time, I'd like to talk about what could be done and what some of these desirable outcomes would be. Could we, for instance, further identify and cultivate beneficial post-fire microbes? I think such a technology would be very useful in mitigating some of the damaging effects of fire. The hope is that with a greater understanding of the post-fire microbial community assembly patterns, we can design microbial inoculation strategies that would increase plant sustainability, forest productivity, water quality, and carbon sequestration, or some combination of all 4, as microbes play a critical role in all of these outcomes. But that being said, we still have a few questions on the basic side of the spectrum that I hope to answer in the next few years, such as how robust are these trends across landscapes and among different plant species, and what are the mechanisms that influence microbiome recovery? With that, I'd like to thank my co-authors, and here's one of them, Chris, and I sampling post-fire with all the necessary PPE, at least in a pre-COVID world. But I also want to express my appreciation to Alyssa Carroll and Don Klingman. Like I was telling Chelsea before we got on, they've really pushed through a lot of the laboratory work while I was out in quarantine, and were able to keep me busy working from home with data generation. My sponsors for this work include the Plant Microbe Interfaces, the Joint Genome Institute, the Southern Sierra Critical Zone Observatory, and the Southern California Edison Fellowship. And I'm happy to answer any questions. Thanks.
Thank you so much, Nicholas. I'm glad, we were able to make it through, and it's super interesting work. It's really fun to hear about sort of, all your body of work on fires over the past few years. So thank you so much. I know I have lots of questions, but I'm going to pause, and if folks want to drop their questions in the chat box, we're a small enough group, and I think we're all internal, and I'm going to open up the chat function. So if you want to post it to everyone so Nicholas can see, or you can post it directly to me and I can read it out loud. You can do it either way. And as folks are getting their thoughts together, I guess I'll kick us off. So have you paired any of your sort of assessment? Like, you've done a ton of awesome work from looking at microbial structure using different like sequencing, or different DNA based approaches. And I'm wondering, have you looked at have you paired that with any assessments of function? Either like in your first study of, I'm thinking, have you paired sort of compared changes in the endophytes or holobiont with plant, success or plant physiology, that kinds of things post-fire? Or then on the flip side, have you looked at sort of how some of these changes in microbial structure in the soil correlate or like in turn influence soil functioning after fires?
Yeah, definitely. So yeah, on the plant side of things, that's definitely something that we would have liked to have done, or something that we would like to do in the future. Because we see these large differences in the leaf microbiome, and we know that the microbiome is really important for the overall health of the plant. But specifically, we want to know if these changes really influence the survivability of these plants post-fire. And so it'd be really interested to go back and back to these plants and see if certain ones survived a year after, or actually at this point it would be almost 2 years after. And it would also be interesting to see if we can bring some of the soil back, and do plants grow better in the soil or worse in the soil? These are all questions that I don't know, we showed that there is a change, but how much does it matter? It's still unclear. On the soil side of things, we did do some sort of different biogeochemical assays and different measurements of nitrogen cycling and carbon cycling. And on the nitrogen cycling thing, we did see that there were large, pretty strong correlations between these different nitrogen cycling functional genes and assays of nitrogen cycling. So for instance, we saw a lot of ammonia oxidation directly after fire, which we also saw when we did our ammonia oxidation assays. And so, we saw both in the genes and in actual function. And so that was pretty cool. We also saw that similarly in the carbon side of things, but to a lesser degree. Part of what we're interested in is we're interested both in the potential that the microbe has and what actually happens. Because if there's a potential there, it should sort of correlate with what actually happens, but not all the time. And so the genetic side of things, all the potential, and so we show that sometimes it correlates well with what's actually happening on the ground, and sometimes it doesn't. But there's so many other factors that influence microbial function other than just your genes.
Mm-hmm. Mm-hmm. Awesome. Thank you for that answer. Mm-hmm. Are there other questions from the group? I know you guys have some. Don't be shy. I'm wondering if you could speak a little bit more about this idea of the sort of inoculation post-fire. So that's something that we think about a lot, actually. We, with the fires that have been sort of sweeping across our landscapes for the past couple of years, and in particular, sort of damaging structures, there's a lot of interest in finding remediation techniques to help with heavy, sort of dealing with heavy metals and, the hydrocarbons, in addition to facilitating sort of recovery of, of plant communities and stuff like that. And so I guess maybe one question to you would be, do you have thoughts on sort of like where you would start with inoculants, like going from commercial through making your own, like broad scale community inoculants to like whole soil inoculants to isolations, that kind of thing? Have you thought about all that? And
yeah, I mean, I have a couple thoughts on that. So, I mean, as I think you're kind of alluding to, is these inoculation strategies don't always work. These are microbial communities have, thousands of different members within them. And so if you're going to add one more species there, who knows if they're actually going to survive in that community. And one of the things that we looked at is, and this is sort of going off a little bit off topic from what you asked, but I think it's sort of germane. And that is, when is the best time to do these inoculations? And there's some work, at least in the human gut microbiome, the best time to do it is when the community is really in flux. So when we have a lot more of that stochastic assembly, it's not necessarily so determined. It doesn't have a whole lot of selection factors on them. And I think one of the cool things is that we see is directly after fire, we do have a lot of stochasticity in the system. And so if you can get out there as soon as you can and put out some of these inoculations, I think it would work better than if you did it a few years down the line. I don't have any data to support that, but that's sort of, if we're taking what we kind of know from the human gut microbiome, I think that, obviously soils are different, but you have to start somewhere. I think that's probably, the place to start with in terms of timing. But in terms of who you put out there, I think, here we show that these environments are really stressful. And so using microbes that have a lot more of these stress tolerant genes would be a good avenue. I'm thinking particularly a lot of like actinobacteria, which have a whole host of other sorts of important functions, but are typically, very stress tolerant. And so, yeah, I mean, I think probably the way to go is to isolate some of these different post-fire microorganisms, look at some of their genes and determine which ones are likely to survive in a stressful environment.
Mm-hmm. Awesome. Thank you for that answer. Yeah. Yeah. It's something that we've been thinking about a lot. We've been sort of poking around with some ideas for doing some work on some science around the sort of post-fire management with inoculants. And we've been thinking about timing a lot too. And when, does it have to go in that same sort of water year? Can it go in the next year? That kind of thing. And that's a really interesting point about sort of when will the community be most receptive almost?
Yeah.
So
I think, I mean, of course, different microbes are probably maybe going to do better at different times, but I think that that's at least, a place you can start. And I don't know if it's true. I mean, that's just what I'm hypothesizing.
So we have another question here. So we have a question from Jay Roberts, who's with Point Blue, and you can, I think, see it. I think you shared it with everybody, but I'll read it out loud as well.
Okay.
It says,
I don't know.
Okay, it'll be in the chat box. I'll read it. So what about soil water content? Yeah. We know that trees can have a huge effect on below ground water, especially large trees, and densely packed forest stands can really lower soil water content. I assume that also affects the microbial community. Is there a potential feedback between the above ground biomass and below ground microbes and water that plays into the patterns associated with recovery from fire?
Yeah, I think that's a huge at least in the aspen study, a huge area that we don't really know much about in terms of that. So we didn't really measure water content. I mean, it's hard to measure water content when you're only going out there a couple of times because water content fluctuates considerably. But I think that Jay is on sort of the right way of thinking about this, in that, of course, plants are going to have a large influence on water content. And I think we kind of got at it indirectly by seeing that, at least in the second study I showed, that the microbiomes of the soil had were influenced by the above ground vegetation. And so I think, a lot of that has to do with sort of the carbon substrates that the vegetation is producing, but also the amount of water that's being transpired and taken out of the soil by the plants too.
Awesome, thank you. Erin Conlisk at Point Blue, she had a question. I'm paraphrasing a little bit, so Erin, correct me if I'm, fill in the details. But the question is generally, do you have thoughts on how your results, like how these kinds of results vary with things like plant type, ecosystems, and fire regimes? So how sort of generalizable do you think these trends are, or are there particular sort of contextual variables that you think will moderate these responses that you're seeing?
Yeah, that's a great question. Yeah, I mean, really, we're just showing kind of two ecosystems here. One is sort of the Sierra Nevada ecosystem, and these, burn at high frequency, or traditionally have burned in high frequency, low severity. And then the aspen system, which is a little bit more of that higher severity, is sort of natural, or at least the historic range of variation is more of a high severity fire regime. And so we do have two different fire regimes here, but like I said, it's really just two ecosystems. And I think fire regime has a lot to do with what we see. So for instance, these in the Sierra Nevada, our work in California, again, like I said, it's adapted to the plants, and presumably the microbes as well are adapted to these frequent low severity fires. Now, we haven't seen those really in many of those areas in at least 50 years. And so instead having these high severity fires come through. And I think that really really makes the system as a whole less resilient, including the soil microbes. And we kind of talked about this in another paper that I didn't really talk about today, but we called these high severity megafires in the Sierra Nevada ecologically novel, meaning that the ecosystems just aren't really adapted to them. And that this idea is that when you have these ecologically novel disturbances, not only do you see a more intense disturbance, but it's also a lot longer time to recovery. And so I think what we saw in the Sierra Nevada, we're going to have a much slower recovery than what we see in the aspen site. But there are a whole host of other types of fires in different ecosystems that I don't know how generalizable we can make it. Some generalizations that we do see is that there are certain microbial taxa that seem to respond really well to fire, or at least increase in their relative abundance compared to the rest of the microbial community, particularly actinobacteria, like I said before, and in particular, the genus Arthropacter does really well even in like boreal forest fires. So I think there's something there, but we still don't know it.
Awesome. Thank you so much for that response and the great questions, y'all.
Yeah.
Are there any other questions? If not, I'll just say, Nicholas, thanks again. Thank you so much for joining us. This was really, it was a treat to get to see you and get to hear about your work. And I'm sure, I speak for everyone when I say, just thank you so much. It was a real pleasure and a great way to spend Earth Day. And thank you so much for concluding this series for us. Super fun last session, and And everyone, thank you for joining throughout this. It's been super fun, and hopefully we can do it again in the future. And yeah, thank you, Nicholas, and thank you, everyone. And we'll see you next time.
Thanks, everyone. Great to see you, Chelsea.
Yeah, you too. Take
Bye-bye.
care.